Bismillahirrahmanirrahim. Assalamu alaikum. Welcome to my YouTube channel, Science for Everyone. Today our topic is related to bacterial cell wall, and the title of my today's lecture is the peptidoglycan structure. We all know that what is basically a peptidoglycan, a layer. It is also known as murine, and it is present in gram-positive bacteria as well as in gram-negative bacteria. Peptidoglycan layer is very much important for the survival of bacteria because it gives strength to the bacteria as well as it gives strength, and also it helps the bacteria to withstand different adverse conditions. Okay, so in today's lecture I will discuss that what is basically the actual structure of the peptidoglycan layer and how it provides strength to the bacteria. And in the next video, I will discuss the biosynthesis of peptidoglycan layer, that how it is formed. And in the next video, I will discuss the role of antibiotic that is used in the destruction of peptidoglycan layer. Okay, so watch all the three videos in a row so that you should be able to answer different questions related to the structure of the peptidoglycan layer, the biosynthesis, and the action of different antibiotics in the destruction of the peptidoglycan layer. In this video, I will discuss the molecular level of the peptidoglycan structure, that what are the different components that are present in the peptidoglycan layer. Okay, so this is very much important for a microbiologist to know about this structure, because when after knowing the structure, he can apply different antibiotics to destroy the peptidoglycan layer. Okay, so let's start with the video lecture. Okay, first of all, you should know about the gram-positive bacteria and the gram-negative bacteria, that how these peptidoglycan layer is present. So this is a structure of gram-positive cell wall, gram-positive bacteria, and this is the cell wall of gram-positive bacteria. In the gram-positive bacteria, it consists of about 45 to 50 sheets of peptidoglycan layer. Okay, so this is the peptidoglycan layer, and after that there is a periplasmic space, and after that there is a plasma membrane. Okay, so if someone asks you that how peptidoglycan layer is present in gram-positive bacteria, so your answer will be that the outermost layer is the peptidoglycan layer, and it is very much thick, about 50 sheets. Okay, and after that there is a periplasmic space, and then there is a plasma membrane. Okay, so this is the structure, the cell wall structure of the the gram-positive bacteria. Okay, after that, if we look to the gram-negative bacteria, so this is the cell wall of gram-negative bacteria. Keep this thing in mind. The outermost layer is our outer plasma membrane or outer membrane. After that, there is a periplasmic space, and then there is a small layer of a thin layer of peptidoglycan layer. It is about 1 to 5 sheets. Okay, and in the gram-positive bacteria, it is about 45 to 50 sheets. Okay, so keep this difference in mind. The difference is also in the cell wall structure, because in the gram-positive bacteria, the outermost layer was the peptidoglycan layer, but here the second layer is the peptidoglycan layer, and it is also in thin in size. Okay, the diameter is not that much larger. Okay, so keep these differences in mind. Okay, because of this peptidoglycan layer, the gram-positive and gram-negative bacteria were divided into two parts. Okay, so this is the peptidoglycan layer. Okay, so I was just showing that where does this peptidoglycan layer exist in the gram-positive bacteria and in the gram-negative bacteria. Now, if we look to the electron microscope image of the peptidoglycan layer in the cell wall, so this is the cell wall of gram-positive bacteria. The outer, if we look to here, start from here, so P stand for the periplasmic space that is present in between the cell membrane and the peptidoglycan layer, as I have shown you in the animated picture. This is the plasma membrane, and this brown color is the cell wall. This is the peptidoglycan layer. As I have told you that it is thick in diameter, about 50 sheets. Okay, so this brown color is the peptidoglycan layer, and it is an electron microscope image. Okay, if we look here to the gram-negative bacteria, So the cell wall of gram-negative bacteria. So the outermost layer is this is the outer membrane. Okay, this OM stand for outer membrane. This PM stand from the for the plasma membrane. Okay, if you look here, so this is the plasma membrane. This pink color. After that, there is a periplasmic space. This P stand for periplasmic space, and after that there is a murine cell wall. Okay, sorry, as I have told you that peptidoglycan is also known as murine. Okay, so there is a the thin layer, this brown color is the peptidoglycan layer. Okay, and after that there is the outer membrane. So just look to the difference, how much difference is in between the gram positive and gram negative bacteria with respect to the location of the peptidoglycan layer. Okay, so I was just showing you the difference in between this. The main aim of our video is the structure of peptidoglycan layer. So let's start with the main structure of peptidoglycan layer. Okay, so if we look here, so this is an animated picture, a very simple picture. In the next slide I will discuss the molecular level. Okay, so if we look here to a small unit, okay, this is 1 unit of peptidoglycan. Okay, so this orange color is the N-acetyl glucose amine, and I will show you the molecular structure of it. And this yellow color is the N-acetyl muramic acid, and just because of it, it is known as murine. Okay, and these four round shaped circle are these are amino acids. Okay, and what are basically these amino acid, I will discuss it in the second slide. Okay, so these four round shaped circles are the four amino acids that are linked to this N-acetyl muramic acid. And after that, if we look here to this bar, so this is the interbridge. It is also known as peptide interbridge. Okay, it is also known as pentaglycine interbridge or peptide interbridge. It binds the layers to each other. Okay, and this is very much important. Okay, just because of this peptide interbridge, these layers are bind to each other, and just because of it, it gives strength to the peptidoglycan layer. So just look here, compare this image with this animated picture. So this yellow color is the N-acetyl muramic acid. Okay, and this orange color is the N-acetyl glucose amine. And just look here to these curved line. These are the peptide interbridge. Okay, these are the peptide interbridge. And look here, this is glycosidic bond. Okay, because these are two glucose molecules, so these are bind to each other by glycosidic bond. Okay, so if someone asks you that how much bonds are there in a peptide, in a single unit of peptidoglycan layer, and what are the bond, so your answer will be that the two glucose molecules are bind by glycosidic bonds. Okay, after that, four amino acids are bind to that N-acetyl muramic acid, and after that, the layers of the peptidoglycan layer are bind to each other by a pentapeptide interbridge. Okay, and this pentapeptide interbridge is very much important because it holds the layers of peptidoglycan layer to each other. Okay, and just because of it, it gives strength to the peptidoglycan layer. Okay, so just compare it with this. These are the peptide interbridge. Okay, and these are the different molecules. Here the amino acids are not shown, but just look here to the, I was just showing the peptide interbridge. Okay, so let's proceed to the molecular level of it. Okay, just look here, this is the molecular level of the peptidoglycan single unit. Okay, if we just take a single unit of this. Okay, if we take a single unit of this. Okay, so this orange color is the N-acetyl glucosamine, and it is a glucose molecule, but it is conjugated with different other components. Okay, different other functional group, so that's why it is known as N-acetyl glucosamine. After that, it is bind to the N-acetyl muramic acid, this yellow in color, by a glycosidic bond. Okay, so if I write GB, glycosidic bond. Okay, after that, this is the glycosidic bond. Okay, After that, it is connected, it is linked to, with 4 amino acids. Okay, the first one is L-alanine, and the second is D-glutamic acid. The fourth one is diamino pamelic acid, third one, and the fourth one is D-alanine. Okay, so these are the 4 amino acids. One thing, keep this in mind, that in gram-negative bacteria, this diaminopamelic acid is present. Okay, this diaminopamelic acid is present, but is in gram-positive bacteria, L-lysine is present instead of diaminopamelic acid. So keep this difference in your mind. It can come in the MCQs, then you should mark the correct option. In gram-negative bacteria, this diaminopamelic acid is present, and in gram-positive bacteria, L-lysine is present. Okay, L-lysine is present in with respect to the diaminopamelic acid. Okay, so this is the molecular structure of a single unit of the peptidoglycan unit. Okay, in the next slide, I will discuss the peptide pentaglycine interbridge, the molecular level. Okay, so again, I am repeating, this is N-acetyl glucosamine. It is bonded to N-acetylmuramic acid by a glycosidic bond, and after that, 4 amino acids are bonded to each other. First, it is binded to the N-acetylmuramic acid. After that, these are bind to each other: L-alanine, D-glutamic acid, diaminopamelic acid, and D-alanine. Okay, so keep this single unit in your mind. How these units combine, so now I will show you. Okay. Okay, now this is another structure of the peptidoglycan layer. As I have told you that I will show you the pentaglycine ring, that how these are interconnected to each other. So this is, as I have told you, this is a single subunit of the peptidoglycan single unit. And after that, just look here. This is a single sub unit, and it is bind to the another subunit by this action. Okay, just look here, this is present in this direction, and after that, the D-alanine of the first unit of the peptidoglycan is bonded to the L-lysine, of the second subunit. Okay, this is, just look here, and it will be present in a upward direction. Okay, inverted direction. For example, if we just write it 1 2, okay, so it will be present in 2 1 direction. Okay, so just because of this direction, they can interconnect to each other. So this is in the case of gram positive bacteria. Okay, and this is the case of the gram negative bacteria. So this is another MCQs. Keep this thing in mind, that the two subunit of the peptidoglycan layer are connected to each other by the help of pentapeptide interbridge in gram positive bacteria. Okay, 5 glycine amino acid connect these two subunits in gram positive bacteria, and in gram negative bacteria, the D-alanine binds to the diaminopamelic acid, forming a peptide bond. Okay, here is a peptide bond. Okay, because when two amino acids are connected to each other, so they are connected to each other with the help of a peptide bond. Okay, so keep this thing in mind. So here, just the D-alanine, the NH, for example, the NH3 NH2 of the D-alanine binds to the, or the COOH of the okay, just looking here, if I write it like this, so the COOH of the carboxyl group of the D-alanine binds to the NH2 group of the diaminopamelic acid, and simple a peptide bond is formed, and they are interconnected to each other. But it only occurs in the gram negative cell wall. Okay, the gram negative bacteria, and in gram positive bacteria, the two subunits are interconnected to each other by a pentapeptide interbridge. Okay, so keep these differences in your mind. Okay, so if we remind now, just look here to this structure. So this was the L-lysine of the one subunit, and this was, if wait a minute. Okay, So this, if you look here, so wait a minute. Okay, so if you look here, so this was the D-alanine and the diaminopamelic acid. So if we compare it to each other, okay, so okay, so this was the L-lysine. Okay, this third one is the L-lysine, and this was the diaminopamelic acid. Okay, sorry, again I want to tell you guys that the third one is the diaminopamelic acid, and the last one is the D-alanine. Okay, so these are interconnected to each other just like this. So by interconnected to each other, they give strength to the peptidoglycan layer. Okay, so this was all about the structure of the peptidoglycan layer. Okay, if you have any question related to this peptidoglycan layer, so you can ask me in the comment box. In the next video, I will discuss the biosynthesis of the peptidoglycan layer, that how these N-acetyl glucosamine and N-acetyl uramic acid come and join to each other. After that, how these amino acids join to each other and then join to the N-acetyl uramic acid, and how this interbridge forms, what are the enzymes that are involved in this this peptidoglycan layer biosynthesis. Okay, and in the next video, I will discuss the antibiotics that are involved in the destruction of the peptidoglycan layer. Okay, so this was all about the today's lecture. Again, I'm telling you that if you have any question related to peptidoglycan layer about the structure, so you can ask me in the comment box. I will answer you there. Okay, thank you very much. God bless you.